I will be discussing sickle cell anemia. Sickle cell anemia is a severe hereditary form of anemia in which a mutated form of hemoglobin distorts the red blood cells into a crescent shape at low oxygen levels. Hemoglobin typically consists of 4 protein subunits, 2 alpha globin, which is produced by HbA gene, and 2 beta globin, which is produced by HbB gene. People with sickle cell anemia have had a mutation specifically in their HbB gene. This mutation changes a single amino acid, causing the amino acid glutamic to become replaced with valine in position 6 in beta globin. This mutation causes the production of a normal version of beta globin called hemoglobin S, which then replaces both subunits of beta globin. The valine causes the hemoglobin S units to stick together to form long-rigged molecules that bend, thus creating the crescent shape of the red blood cell. This was seen in the previous slide. On the right, you see a normal red blood cell compared to a crescent-shaped sickle cell on the left. These crescent-shaped cells die prematurely, which causes anemia. In 1910, physician James B. Herrick published a description of sickle cells in a blood sample from a 20-year-old African-American male. In 1933, scientists tested 2,500 African Americans in Memphis to determine if sickle cell trait and sickle cell disease were separate. In 1934, the sickle cell crisis was suggested to result from the blockage of small blood vessels. In 1940, scientists believed sickle cells were created by the exchange of oxygen for carbon in small blood vessels. In 1948, National Heart Institute granted Dr. James Neal to study the disease to suggest sickle cell was caused by a high level of fetal hemoglobin in the red blood cells. In 1949, Dr. Linus Pauling reveals the cause was due to abnormal hemoglobin protein molecule, thus giving its name molecular disease. In 1949 through 1950, scientists found that sickle cell trait from both parents caused the disease, while the trait from only one parent caused the trait. In 1951, Linus Pauling's story raised public awareness of sickle cell anemia. In 1953, a diagnostics tool was developed to identify sickle cell disease. In 1955, a blood test was later developed. In 1957, scientists showed the abnormality of sickle cell hemoglobin was due to amino acid substitution in protein, therefore becoming the first genetic disorder whose molecular basis is known. In 1959, Marclan A. Walker raised awareness by publishing Life with Sickle Cell Disease in Ebony magazine, which was then later published in Time magazine. In 1963, a three-dimensional structure of the hemoglobin protein was deciphered using X-ray crystallography. In 1971, National Association for Sickle Cell Disease was founded. In 1973, national screening methods using blood spots on paper was later created. In 1975, New York became the first to require newborn screening for sickle cell. In 1980, scientists discovered that sickle cells were stickier than normal red blood cells. In 1996, maternal blood sample for parental diagnosis was created. In 1998, the FDA approves a hydroxyurea for sickle cell disease treatment in adults. In 2002, Health Resources and Services Administration program started. Here are a picture of Linus Pauling, James Neal, and James B. Herrick. Sickle cell anemia is found throughout the world, mostly in people of African descent. 70,000 to 80,000 Americans. The disease is estimated to occur in 1 in 500 African Americans and 1 in 1,000 to 1,400 Hispanic Americans. It affects approximately 100,000 Americans today. 1 out of every 365 Black or African American births, 1 out of every 16,300 Hispanic American births. This disease affects the liver, heart, kidneys, gallbladder, eyes, bones, and joints. The lungs, spleen, and also brain. And this disease can last lifelong. Signs and symptoms: anemia, episodes of pain, painful swelling, infections, delayed growth, vision problems, dizziness, fatigue, low oxygen in body, and malaise. The questions that I have is, why is this mutation prominently in African Americans? And my second question is there some way that